Today our topic is gene technology. We will start with what is genetic engineering. Genetic engineering is combining genes, and by genes I mean the DNA, from two or more different organisms. For example, there are many applications, including creating genetically modified organisms. For example, you could take a gene from a salmon, a fish, for being resistant to cold, and put it into a strawberry plant. So that strawberry was resistant to cold when it grew. The strawberry wouldn't taste like a salmon. It would just have one piece of DNA from the salmon put into the strawberry. So it would have its normal strawberry DNA plus this one piece of DNA from the salmon. And now when it grew, it could grow in cold environments. Another application would be vaccines against viruses. Another application is DNA fingerprinting, which we'll talk about in just a second. Another application is the Human Genome Project. As you can see from this slide, here's a piece of bacterial DNA, here's a piece of human DNA, and we've joined them together. That is genetic engineering, taking DNA from two different species and joining them together to create something called recombinant DNA. Again, applications: creating strawberries that are blue or that are resistant to cold, creating bigger mice or bigger cattle for eating, creating drugs and vaccines for DNA fingerprinting. One really interesting example is to take a piece of DNA from a jellyfish that makes them glow and to put that DNA into organisms like mice or pigs or rabbits. Not only is it cool, it also lets you know whether or not your experiment worked, because if they glow, then they did get the DNA from the jellyfish. The scientist who discovered this GFP or glow gene won the Nobel Peace Prize for it in the 90s. DNA fingerprinting is an application of gene technology, where DNA fingerprinting is cutting DNA into pieces and then loading it into a gel and separating the pieces based on size. And there's a video you will watch that goes over this process. Here is a finished DNA fingerprint. There are two applications for DNA fingerprinting that we'll discuss. The first one is a crime scene investigation. Let's say that you collected DNA from a crime scene. Here's the crime scene DNA. You then used special DNA scissors called restriction enzymes to cut that DNA up into pieces. That DNA would be injected into this part of the gel. Electricity would run through the gel, forcing the DNA to all move in this direction. Smaller pieces of DNA would go farther in the gel, and bigger pieces of the DNA would get stuck at the top of the gel. So what would happen is the DNA would be separated by size. And this is called the DNA fingerprint because it is unique to this person found at the crime scene. Then you have 3 suspects, and you collect their DNA. So here's suspect 1's DNA. Here's suspect 2's DNA. And here's suspect 3's DNA. Again, you cut up the DNA using the DNA scissors, load it at the top of this gel, run electricity through the gel to see how the DNA bands separate out. Smallest pieces are at the bottom, biggest pieces are at the top. Now for crime scene, you need to see which suspect is an exact match for the crime scene DNA. So it looks like the crime scene DNA and suspect 2 are exact matches, which means suspect 2, this is their DNA because it is an exact match. So which suspect was at the crime scene? Suspect 2. How did you determine that this suspect was at the crime scene? Well, suspect 2 and the crime scene DNA are identical. So the crime scene DNA and suspect 2 have identical or the same DNA fingerprints. So that's how you would identify crime scene DNA. You would match exactly the suspect with the crime scene. There is another use for DNA fingerprinting. That's to determine paternity. Who is the father of a child? Below, this DNA fingerprint shows the mother's DNA. So this is the mother's DNA in lane 1, and I always label them to remind me who is who. So this is the mom's DNA. In lane 2, you'll find the child's DNA. Lane 3 shows you suspected father A's DNA. And lane 4 shows you suspected father B's DNA. So to use DNA fingerprinting on paternity, the first thing you need to do is say, okay, which bands of DNA from the mom did the child get? So basically this child got 50% of its DNA from its mother and 50% of its DNA from its father. So when you look at the DNA bands, all of the bands that the child has must match up with the mother or the father. So the way I do this is I first take the child and I look at each band. Okay, this child could have gotten this band of DNA from the mother. This band, however, does not match with the band on the mother, so it must come from the father. Let's keep going. This band that the child has is an exact match with the mother's DNA, as is this band. However, this band does not match with the mother. So these are the 2 bands of DNA that must have come from the child's father. If we look, lane 3, father A does not have these bands. However, lane 4, father B does have these bands. So the suspected father in this case must be father B. You can see the child also could have gotten some of the other DNA bands from the father. So the first question over here is, what genes do the mother and the child have in common? Okay, and we can look at the bands. We've labeled them with letters over here to represent the different genes or pieces of DNA. So the mother and child have this band in common, band A. They have this band in common, band F. And they have this band in common. I can't tell if that's G or H. It doesn't really matter. We'll say H. And then who is the father? Suspect A or suspect B? We know that suspect B is the father. And how did you determine this? Because any of the child's bands that don't match the mother match with suspect B. So unlike in crime scene, the child's DNA is not going to be an exact match with the mother and the father, but it's going to be a mixture of the mother and father's genes. So what should you do with this information? First, you'll fill in the notes on Google Classroom, and these notes match up with what I just said. Next, you'll watch an Amoeba Sisters video on gel electrophoresis and answer these questions below. Finally, you'll watch this video introducing genetic engineering and answer these questions. And then you'll submit your work in Google Classroom. In class, we'll discuss your work and we'll go over next steps.